Hello, everyone, and thank you for joining this latest Science AAAS webinar, Deciphering Aging, Linking Senescence with DNA Damage and the Cell Cycle. I'm Sean Sanders, Senior Editor for Custom Publishing at Science, and I'm very happy to be here to moderate today's webinar broadcast, the third in our Deciphering series. You can access the first two parts by clicking on the links just below the event title. And please do look out for our final installment in this series on November 14th of this year. Today we're going to be focusing on aging, cancer, and cellular senescence. Senescence describes the complex cellular response to stress that includes irreversible arrest of the cell cycle and thus prevention of the proliferation of defective or damaged cells. This effect makes senescence a key component in the body's tumor suppression response and initialization of repair pathways, providing a health-promoting mechanism. Conversely, senescent cells can accumulate in the affected tissues of persons with age-related diseases such as dementias, arthritis, atherosclerosis, and others. Such accumulation is considered a hallmark of aging that drives many age-related pathologies. These seemingly contradictory roles make cellular senescence an interesting research target for developing cancer suppression therapies, as well as improving health maintenance and extending human lifespans. I'm delighted to welcome our two speakers for today's presentation. They are Dr. Sheila Stewart from Washington University in St. Louis, Missouri, and Dr. James Kirkland from the Mayo Clinic in Rochester, Minnesota. Thank you both so much for joining us on the line today. Before we get started, I have some important information for our online viewers. If you take a look at the top right of your screen, you'll find photographs of today's speakers as well as a View Bio link that you can click on to read more details about their background and research. Underneath the slide viewer is the Resources tab, where you can find additional information about technologies related to today's discussion, as well as a PDF of the slides. After the speaker presentations, we'll have a short Q&A session during which we will address some of the questions submitted by our live online viewers. So if you are joining us live, please start thinking about some questions now and submit them at any time by clicking the Ask a Question button below the slide window, typing the question into the message box, and clicking OK. You can also log into your Facebook, Twitter, or LinkedIn accounts during the webinar to post updates or send tweets about the event. Just click the relevant icon at the top left of the screen. For tweets, you can add the hashtag #ScienceWebinar. Finally, thank you to Cell Signaling Technology for sponsoring today's webinar. Now on to our first speaker for today, Dr. Sheila Stewart. Dr. Stewart is a professor in the Department of Cell Biology and Physiology at Washington University School of Medicine in St. Louis and Associate Director for Basic Science at the Siteman Cancer Center. A recent focus of her laboratory is examining how age-related changes in the pre-metastatic niche facilitate tumor cell seeding, dormancy, and outgrowth in the bone, and how these changes alter the local immune response to facilitate tumor cell proliferation. Welcome, Dr. Stewart, and thank you so much for making the time to be with us today.
Thank you, Sean, and thank you for the invitation. What I'd like to do today is share some of our research that has been focused on trying to understand how age-related changes, specifically in the stromal compartment, contribute to tumorigenesis. And really our interest in this derives from the fact that as you get older, you see more cancer. And so, of course, when one looks at data like this, one has to ask the question, why is it that we see basically no cancer on a human population scale until the 5th or 6th decade of life? And I think, of course, the answer is very complicated. Cancer is complicated, but I think many of us would agree that the bulk of human cancer is the accumulation of cell-autonomous genetic mutations. And in the face of ongoing and robust DNA repair mechanisms, it just takes time, not only for genetic mutations to accumulate, as we know they do with age, but for the right combination of those mutations to arise within a single cell. When one looks at this, we have to then ask a question, is that single variant sufficient to drive this rapid increase in tumorigenesis that we see in the 5th decade of life? And we've really been focusing on asking the question, is it possible that age-related changes in the surrounding stromal compartment, in the non-cancer compartment, if you will, would also contribute to this increase in that tumorigenesis in the 5th decade of life? And we've really focused on the possibility that the accumulation of senescent cells play a part in this rapid increase. The reason that we've been interested in this is, first of all, we know that these cells do accumulate as a function of age in every tissue in the human body that's been looked at. But really more importantly, Judy Campisi was actually able to show almost 15 years ago now that senescent stromal cells are able to recapitulate nearly every phenotype that has been ascribed to cancer-associated fibroblasts. So these are fibroblasts that are typically found inside an actually growing tumor. They are not cancer cells themselves, they're fibroblasts associated. And what Judy Campisi was able to show is just as has been described for these cancer-associated fibroblasts or these CAFs, these cells can directly drive tumor cell proliferation, they can drive migration, invasion, angiogenesis, and they can also alter the local immune response, really suggesting that these cells would create local environments that would support the outgrowth of actual tumor cells. Now, I think some of the best data to really support this idea was a paper that was published by Van Dersen's group actually a couple years ago using a mouse called the ink attack mouse. And so this is actually data from their Nature paper. And I don't mean to go into the genetics or how this mouse works. It certainly is a good read if an audience has time to look at this paper. But I think what's important here is if one looks, when one supplies an AP drug, you basically kill every senescent cell in the body of that animal. So what they were doing in this particular figure was simply asking the question, if we look at spontaneous tumor rates inside mice, do the presence of senescent cells have any influence in the emergence of those spontaneous tumors? And what you can see is whether you're looking at males or females, whether you're looking on a mixed background, which is that left graph, or the pure background, which is the right graph, elimination of senescent cells is able to specifically delay the onset of spontaneous tumor rates. If any of the audience has questions as to why that might be, I think the answer itself might be complicated, but I think it does raise the very important possibility that the accumulation of those senescent cells, which are being eliminated in those mice, is in fact influencing the outgrowth of nascent tumors. Now, of course, the question then is, if we do accept the possibility that these stromal cells can drive tumorigenesis, how do they do it? And we like to say in the lab, they basically make everything yummy you can imagine if you're a tumor cell wannabe. They are dumping out large numbers of mitogens to drive proliferation. They dump out enzymes that modify the extracellular matrix, another component we know is important for transformation. And they actually dump out a large cadre of immune modulating enzymes, cytokines, and inflammatory cytokines as well, remembering that chronic inflammation actually is the second largest risk factor for the development of cancer, and these guys are able to make a lot of these cells, or a lot of these chemokines, excuse me. And so if you then look at both those CAFs and those senescent cells, all of those features that we associate with them could be ascribed to a number of these different molecules. And so one of the things that we started asking is it possible that these senescent cells, in fact, modify the local environment? And it's interesting that if you actually look in human skin, so here we're simply looking at aged skin. We're either going to collectively call it young if it's under 30, which is shown on the left side, or aged if it's over 60, showing there on the right side. And what we're showing here, and this is a recent publication from our group, is co-staining with P16, which is accepted as a reasonably good marker for senescent cells, and CD45, which is a pan-leukocyte marker, a marker that allows us to look at immune cells. And what was interesting, and this has been reported by others as well, that as you see an accumulation of senescent cells, more P16 positive cells in those aged skin versus the younger skin, we also see an accumulation of CD45 positive cells. In other words, there are more immune cells in the skin of these individuals. And that's recapitulated if we actually look in mice. So showing here we can actually use another stain called SA-beta-gal that's shown on the top set of slides. We're also looking at immune markers on the lower panel there in the IF. I think it's easier if the audience were to just focus their attention over to the graphs on the right. And basically, perhaps not surprising, what we can show is older mice. These are mice that are 24 months old, 2 years of age, have significantly more senescent cells in their skin relative to a 2-month-old mouse, and similarly, they have more immune cells. Again, this isn't surprising. This is what we might have expected based on what's been published by many groups around the world. And so with this information in hand, we wanted to ask a question. Would it be possible to create a genetic model in which we could age, if you will, the stromal compartment in an otherwise young mouse and ask how that might actually affect tumorigenesis. And so we did this. I'm not going to go too much into the genetics. We published this late in 2016, so certainly the audience can look to those papers and look at how the genetics were set up. But suffice to say, using a collagen promoter, something that's not expressed in the epithelium, we were able to drive a tamoxifen-responsive Cre-ER construct in addition to a conditional allele of a mouse that's expressing P27 upon the supplying of tamoxifen. Why did we choose P27? In all honesty, because it works. We were able to show, and this is all in supplemental data, of course, that when we overexpress P27 within the cells of these animals, basically we could activate senescent phenotypes. And trying to actually convince you of that, I'll show a little bit of that data here. And so what you can see here now, looking at age-matched mice, we're looking at litter-mate controls as well as what we ended up calling the FAST mice for fibroblasts accelerate stromal-supported tumorigenesis. And basically what you can see, again, looking on those top panels for that SA-beta gal, that senescence marker, when we activate senescence using our conditional allele, we do in fact see more senescent cells. That's shown on the graph on the right. And again, when we go and look and count CD45 positive cells, so these are, again, immune cells, that's that graph on the lower right. In the FAST animals, we actually see significantly more immune cells than we see in the control animals. And we thought, okay, this is very interesting. We are able to actually recapitulate what we saw in those older mice, what we were also seeing in older individuals, by simply activating senescence exclusively within the stromal compartment. And when we started to carry out flow cytometry approaches to try to get an idea of what these cells look like, we were actually a little bit frustrated. And at first, we didn't actually see significant increases in total CD45 cells when we looked at the skin at large. And so we started going back and looking a little bit more carefully at what the skin actually looked like. And what we were able to actually show, again, here we're now switching to an IF figure instead of the IHC that I had just shown on the previous slide, where we again went in and now we're looking for CD45 cells. Those are the immune cells. What I failed to mention is our genetic model also has a lineage tracer. So all senescent cells in which we've activated our allele are also GSP positive. We actually noticed that it looked as if the CD45 cells were often encircling right next to our senescent cells. And of course, when we went in and quantitated this, again, looking at association, this is shown here on this slide, basically a negative number would be a repulsive event, if you will, and a positive number is an attractive event. What you can see is if you look at the ability of CD45 cells to associate with the surface of the epithelium or even with other CD45 positive cells, it really was a neutral event. But when we looked at the ability of CD45 cells to specifically associate with our senescent GSP positive cells, we did see a significant increase in that association. And what was interesting is when we went back to the skin of our either younger cohort or more aged cohort of humans, we saw the same association. That is to say that the P16 positive senescent cells associated with CD45 cells much more significantly in the older individuals than in the younger individuals. And so thinking about this association, I'm not going to go through the data. This is already published, and I'm going to just summarize it in the next slide. But basically what we were able to show was that the senescent cells, shown as, blowing up cells, if you will, in this diagram, secreted a large number of factors. I've shown you some of those factors already. And what was interesting is that these factors were sufficient to recruit immature granulocytes that actively were able to inhibit CD8 killer T cells. And our hypothesis is that if this is actually happening in the human skin, certainly we can show that the senescent cells are there, we can show that the immune cells are there. The hypothesis is perhaps this is creating a localized suppressive environment that, if you will, gives a initiated cell, a cell that's on the road of transformation, has some of those mutations, some space to breathe, if you will, to actually accumulate additional mutations without being attacked by the immune system. And perhaps this is a place in which we could see the emergence of a tumorigenic clone, if you will. And so that's where we've continued to focus our attention now, looking at the primary site and asking how these senescent cells are mechanistically calling in these immature granulocytes and affecting the immune system. However, this was not the only thing that we wanted to do with these mice. We also wanted to ask another question. And in this system, we actually wanted to ask the question, is it possible that senescent cells that, again, are accumulating stochastically as a function of age also have the capacity to modify, if you will, the pre-metastatic niche? And so what I'm showing you here up in the upper right is some staining, again, for that P16 in the actual bones of humans. And we're doing a co-stain with IL-6. This is one of those many factors that we often associate with senescent cells. So certainly we can see in the bone, as has been reported in nearly every other tissue in the human, senescent cells are definitely there. And so, of course, the question then, as I just said, was to ask, do these cells that we know can accumulate as a function of time change the niche? And if they do, is it possible that those alterations, if you will, do in fact seed tumor cells? And why is this potentially important, and why were we focusing on the bone? Well, we've spent a significant amount of our time focusing on breast cancer, and bone is in fact the most significant site for metastasis. And so the idea here is perhaps these alterations in the pre-metastatic niche, in fact, do end up being areas of seeding. Now, I think this is conceptually a very important way to think about this problem, because there's been some amazing work that's come out over the last several years from other groups that have made the argument, and I think the data is quite strong in this case, that primary tumors, so in this case we're saying breast cancer, but you could put any other cancer name in this place, primary tumors are able to release factors that systemically mobilize bone marrow-derived cells, move them into the pre-metastatic niche, whether it be the lung or other niches that have been looked at, and really fertilize that niche, if you will, create an environment that really is susceptible for the outgrowth of tumor cells upon their arrival. And I think the idea that we're putting here is a little bit different, in that one does not necessarily need to evoke the presence of a activating event, if you will, from an actual primary tumor, that it might be just as likely that things that happen as a function of age are sufficient to change that niche and create a place in which these metastasizing tumor cells can seed. So again, we turn back to the mouse that we had created, and this was a bit of fortuitousness on our part. This collagen Cree that we had chosen to use is actually able to activate specifically in osteoblasts within the bone. And for the purposes of this discussion, while there are many important cells within the bone, we really just need to think about two of them. One is the osteoblast, which makes our bone, if you will, the other being the osteoclasts, which eats our bone. And in a well-working bone, if you will, they live in perfect harmony, just enough making, just enough eating. And so what we were able to show is upon tamoxifen, or activation of our transgene within the bones of these animals, we could show that our transgene was in fact active. This is an IF. I hope it shows well on the screen for the viewers, but basically what I'm showing here is our GFB lineage tracer, which marks our senescent cells, corresponds with expression of osteocalcin, which is a protein that is highly expressed by osteoblasts. So certainly we were able to activate these cells within our bones. If we took a look at what was happening to those osteoblasts, looking at them in vitro here, what we could show is by adding tamoxifen, our activating gene, we saw significant increases with, again, that senescence-associated beta-galactosidase. This is one of the markers that we prefer to use when we're looking at senescent cells. And associated with the activation of senescence was, again, the expression of all of that yummy stuff. Here I'm simply showing you IL-6, which is one of those senescence-associated secretory phenotype proteins, or we also call it a SASP protein. Similarly, when we looked inside the actual bones of these animals, we could actually see, again, activation of senescence-associated beta-galactosidase. So the pictures are on the top, but I'll just draw your attention for the ease of talking about it to the graph below. And again, if we're looking at littermate controls and age-matched fast mice, inside specifically the bones of these animals, we saw a significant increase in, again, the senescence-associated beta-galactosidase. Again, going back to the IF, using our lineage tracer to identify those senescent cells in the bones, we could also see that not only in vitro were these SASP factors expressed, but also in vivo. Again, I'm only showing IL-6 for ease of discussion. So again, arguing that inside these animals, upon supplying tamoxifen, we activate our transgene, we activate senescence specifically within the bone in osteoblasts, and that leads to the expression of senescence-associated secretory phenotype proteins. IL-6 in this case. And so this put us in a position then to ask, have we altered the pre-metastatic niche, in this case the bone specifically, and does this alteration create an area in which these cells can actually grow? So unfortunately, when it comes to the mouse and working on bone metastasis, there is no spontaneous model for bone metastasis. So what we decided to do was to actually turn our attention to a syngeneic model, which would importantly allow us to look at seeding into a metastatic site, again in this case the bone, in a synchronous manner. And for these types of studies, many people in the field, including ourselves, prefer to use an intracardiac infusion, basically allowing one to bypass the lungs and allow the bones to actually be seeded. And so we did this experiment, supplied breast cancer cells to these animals, and asked, what do we see? Do we see an increase in metastasis in the bones? And as this graph is showing here on the right, that is exactly what we saw. We saw a significant increase in metastases within the bone. And of course, if there's time at the end, I'd be more than happy to talk about why we actually saw a bimodal distribution, which I think is quite interesting. But in the favor of time, I'm just going to show you basically what we were able to show in this particular system and what our model is. So if one looks at a normal bone, I think I've already mentioned for the discussion today, osteoclasts eat your bone, osteoblasts make your bone, and they live in perfect harmony. They actually talk to each other. However, in our system, we were able to specifically activate senescence within the osteoblasts of these animals. We could show that they would express these senescence factors, including IL-6. And this actually, we were able to show increased localized osteoclastogenesis. So we actually had more bone turnover locally around these activated senescent osteoblasts. We were also able to go on and show that this resulted in increased seeding specifically to these areas of bone turnover where we had our senescent osteoblasts. And this is where the outgrowth of our tumors were growing. We also were able to go on and show that the major driver of this increase in osteoclastogenesis was in fact IL-6. In fact, it was already known in other systems that if there's enough rank ligand around, which is what is important for driving osteoclast differentiation, IL-6 can actually enhance this. And we were also able to show that if we blocked IL-6 in this system, we were able to significantly reduce the amount of metastasis that we saw in these animals. Now, in the last little bit here, what I'd really like to share with you is a news story in which we've taken this information and really related it back to studies we had carried out several years ago and used this to our advantage to start looking at therapeutic avenues in which we might be able to limit these cells. And so IL-6 is something we have been thinking about for a long time. In fact, several years ago, we had published a paper in which we've been able to show that many of these SASP, or senescence-associated secretory phenotype proteins, are also expressed, as you might not be surprised, in these casts, in the stroma of breast cancer patients. And what was interesting for the particular study that I'm mentioning here is we were able to show there was an overlap that a large number of these particular factors were responsive to the p38 stress kinase. And we were able to go on and show that, in fact, the way in which activation of p38 is able to increase the expression of these SASP factors is through stabilization of their messenger RNAs. And in fact, we were able to show a couple years ago that, again, trying to go back to studies Judy Campisi had done many, many years ago, if one is to take just syngeneic cells and basically mix non-senescent or senescent fibroblasts with poorly tumorigenic cells, inject those into animals, one sees a significant increase in the growth of these tumor cells. So we're looking at this green bar versus the blue bar. Importantly, though, if we went back to those same fibroblasts and we used CRISPR technology to remove p38 from those fibroblasts, what you can see in those orange bars, looking at two different clones, one completely loses the ability of these senescent fibroblasts to increase the growth of these tumor cells. Again, this is in a primary setting. We also were able to show that we could use drugs to therapeutically target this. So if we took those same fibroblasts that were able to support the increased growth, that's actually the bar that goes up higher in the graph, and we were supplied a orally available p38 inhibitor, we could completely eliminate that increase in tumor growth that we saw. And so really this began getting us to start thinking again about the commonalities that one sees between senescent stromal cells and activated stromal cells, or CAFs. And we wanted to start asking the question, is it possible that we could use the approaches that we've uncovered studying senescence to more broadly target activated stroma? And I had decided to just throw in this paper from Bernica a couple years ago showing that, in fact, in other systems, not just looking at breast cancer, but in primary lung tumors, again, P38 within the stromal compartment plays a very important role in stimulating primary tumor growth in this particular model. So we moved back to our bone model. Again, we wanted to ask the question, could we target activated stroma in the bone? And again, we needed to deliver the tumor cells to the bones of these animals. So to do this, we took a breast cancer line, we introduced it into the intercardiac space of these animals, waited 2 days, and then randomized our animals onto either what I'm going to call in this particular discussion standard of care. So this is a chemotherapeutic approach. This is paclitaxel that is actually used in the clinic, or into a P38 inhibitor that we had available through our partners at Confluence Life Sciences. And what I'm showing you here is what in fact happened when we looked at the bone. Again, this was our expectation, right? That perhaps if we targeted P38, we could in fact limit tumorigenesis. And so what one can see is basically looking at the graph, you can see you get distributions of tumor cells that are significantly reduced when mice are randomized onto paclitaxel, and we see exactly the same reduction in tumor growth when we put these animals on P38 as a single agent. And then if we went on, we actually got, I think, what I would call bonus. We saw a very similar effect when we looked in visceral organs at large. Again, P38 as a single agent was sufficient to limit tumor growth, and it was equivalent to what we saw with chemotherapy or paclitaxel. Now, one might say at this juncture, that's great, it's interesting, but mechanistically, that doesn't really argue that you're affecting the stromal compartment in any way. In fact, you've orally dosed animals, and all the cells are going to be bathed in these inhibitors or in the chemotherapy. And so we went back first just to look in vitro, and what I can show you here is, as expected, these particular tumor cells are in fact sensitive to paclitaxel. That would be the blue bar. They don't care one bit if we put our P38 inhibitor on them. And I think perhaps more persuasively, if we took these exact same cells, and here we're using shRNA approaches, we limited P38 expression, again, introduced either the P38 limited cells that were depleted of P38 versus wild type cells. And as you can see in the graph that I'm showing here, these tumor cells grew just fine. They really didn't care at all that they had lost some of their P38 expression. So this was interesting to us. However, we had some concerns with this particular approach, one of which is there's been some really great data from Christopher Klein as well as Julio Guizzo and others that P38, in fact, plays a very important role in maintaining tumor cell dormancy. And so, of course, many of our clinical partners have gotten quite concerned about this idea of using P38 to specifically go after the stromal compartment if, in fact, one might affect the ability for dormant cells to remain dormant. However, it's important to note that the studies, particularly from Julio Guizzo, have argued that this induction or enforcement of dormancy is apparently running through the ATF kinase, whereas the stabilization of these senescence markers that we had been focusing on was mediated through the MK2 pathway. And so we decided, again, working with our partners at Aclaris Therapeutics, that perhaps we could move farther downstream, closer to that mRNA stabilization, and again, use MK2 inhibition to affect tumor growth within the bones. So here what I'm showing you is data I hope can convince the audience that these cells don't care if we inhibit MK2. Again, in vitro data is shown on the left where cells bathed in the MK2 inhibitor grow just fine, similar to the cells that are in vehicle. But more importantly, if we look to the right here, we can see that shRNA depletion of MK2 has no effect on the ability of these cells to grow within the bone. And so these cells grow just fine. Again, when we put our animals on these inhibitors, then what we can show, though, similar to what I showed you for the P38 data, if we limit MK2, both bone metastasis and visceral metastasis are significantly reduced in this model, and this does translate to a small increase in overall survival. And so in the last 2 minutes, what I'd like to show you is really the bonus we got from this. I like to say it's a twofer. We got 2 for 1. And what is this I'm talking about? Well, one of the major problems that patients have once they have metastatic disease to the bone is this perfect balance between osteoblasts and osteoclasts become disrupted. Many people in the field like to refer to this as the vicious cycle. Basically what this is tumor cells arrive in the bone. They actually drive increases in osteoclastogenesis, which releases a variety of growth factors that are locked within that bone. And that in and of itself is sufficient to drive further proliferation of tumor cells. And this, of course, is not without significant consequence. Indeed, one of the largest problems for patients suffering bone metastasis is a significantly high risk for fracture rates, which significantly will limit patients' ability to get around, quality of life, as well as progression of disease. Now, some of this has actually been fixed by the advent of the bisphosphonates, the ability to actually poison these cells, and in fact, this has significantly reduced many skeletal-related events in these patients actually by a third, again, regardless of the type of tumor we're talking about. And the way in which this works basically is the osteoclasts become poisoned as they attempt to eat through the bone. And so we actually wanted to ask the question, could we affect bone quality? And the reason that we decided to focus on this is it had already been known in the literature that the P38 pathway, MK2 specifically, plays a very important role in bone quality. In fact, if you limit if you supply P38 inhibitors or MK2 inhibitors, you can limit both osteoclastogenesis as well as the ability of active osteoblasts to resorb bone, which is basically the PIT data that I'm showing in the lower right corner. And so basically when we went back to ask what was happening to the quality of the bones of these particular mice, what I'm showing you here in this last bit of data slides, again, we're looking at trabecular BVTV. You can just think of it for the argument today as density. A normal mouse, that's the red, non-tumor bearing mice, would have a BVTV of about 0.3. I can't show you the data from mice on vehicle alone because their bones are so destroyed, we can't even measure the BVTV, so I've put imaginary data down there as that big orange oval. But basically what I'm showing here is that when you put animals on standard of care, paclitaxel, we can, in fact, limit tumor growth. I've shown that to you already. But we significantly affect the BVTV or the bone density. However, both P38 and MK2 as single agents significantly protect the bones. And this is important because, and different from what we see with the bisphosphonates or zoledronic acid, as has been shown by many groups before, supplying zoledronic acid, this is the green circles, does in fact protect the bone density, but it really has no ability to limit metastasis or tumor growth, I should say. So unlike zoledronic acid, our approaches with P38 and MK2 are both limiting tumor growth and protecting the bones. And so as I said, this really qualifies as our twofer, if you will. I think that this approach is limiting the ability of the stromal compartment, whether we're talking about activated stroma or senescent stromas, ability to directly fuel tumor cell growth, but it's also protecting the bones by limiting osteoclastogenesis. And with that, I'd like to actually just finish with the most important slide, which are the people that have done all of the work. They're listed here, as well as our collaborators and our funding, and I'd like to turn it back over to Sean.
Wonderful. Thank you so much, Dr. Stewart. We're going to move right along to our next speaker for today, Dr. James Kirkland. Dr. Kirkland is a board-certified specialist in internal medicine, geriatrics, and endocrinology and metabolism, director of the Robert and Arlene Kogod Center on Aging at Mayo Clinic, and the Nobel Foundation Professor of Aging Research. His research covers cellular senescence, age-related adipose tissue and metabolic dysfunction, and development of agents and strategies for targeting fundamental aging mechanisms to treat age-related chronic diseases and disabilities. Dr. Kirkland published the first article about drugs that clear senescent cells, known as senolytic agents. Thank you very much for being on the line today, Dr. Kirkland.
Thank you very much, Sean, and thanks, Sheila, as well, for the excellent discussion. I'm going to speak about senolytic drugs and their potential path towards translation. By way of disclosure, Mayo holds patents that are licensed related to senolytics. I've had no payments personally so far, but I may be paid in the future. So there are a number of processes or a number of stimuli that can impact the rate at which age-related changes occur. Things like obesity, radiation exposure, exposure to chemotherapy drugs, and all kinds of other endogenous processes, including wear and tear, genetic issues, the growth hormone axis. So there's a very complex set of factors that can drive the rate at which aging changes occur in people. Also, downstream of fundamental aging mechanisms that these endogenous and exogenous factors drive are many effects of the fundamental aging processes. These include multiple age-related chronic diseases. So virtually all of the major chronic diseases, chronologic age is the biggest risk factor for. For example, your risk of having a heart attack is increased 2 to 4-fold by having a high cholesterol, by smoking, by having a positive family history, by having high blood pressure. But if you're 85 as opposed to 30, your relative risk of having a heart attack is increased 1,000-fold. So there are a couple of hundred major chronic diseases where chronologic age itself accounts for somewhere around half the risk. Then there are also the geriatric syndromes. These are things like frailty, age-related muscle impairment or sarcopenia, falling, immobility, mild cognitive impairment, and others. And another set of things which occur in advanced age are related to decreased resilience, that is, decreased ability to respond to a stress. For example, older people take a longer time to recover after pneumonia or surgery than other people and have a poorer response, for example, to immunization than younger people do. So the question is whether it's possible to target the things that change with aging at some point. And a good potential intervention point, a nexus point for interventions is potentially fundamental age-related mechanisms that cause these downstream effects of aging. And different groups have categorized fundamental aging mechanisms into 4 to 13 sets of processes. I like to think rather simplistically of 4 sets of processes, one of them being chronic sterile low-grade inflammation associated with fibrosis. This occurs in advanced old age, in tissues where phenotypes develop, but also occur in the context of chronic diseases or that at the sites of etiology of those diseases earlier in individuals who contract those diseases. Macromolecular dysfunction and organelle dysfunction is another broad category, and this includes things like telomere shortening, single-strand breaks, transpositional events, and other things that affect DNA structure. Changes in proteins, including aggregation, failed autophagy, misfolding, changes in carbohydrates, including formation of advanced glycation end products, as well as dysfunction of various kinds of organelles, most notably mitochondria. And then there are changes that occur in stem and progenitor cell function that contribute to both what occurs in elderly individuals as so-called aging phenotypes and also occur at the sites of etiology of age-related chronic diseases. And then the fourth process, and the one that I'm going to focus on, is cellular senescence. All of these 4 processes are interlinked. It tends to be the case that if any one of them is active, the others are as well. And if any one of them is targeted, the others tend to be targeted also. So as we heard in the last talk, cellular senescence is a complex process. There are perhaps 30 or 40 inducers of cellular senescence. Broadly, these include DNA damage and other kinds of oncogenic signals, as well as effects of reactive metabolites, repeated cell division and exposure to mitogens, proteotoxic stress, including protein aggregates, and I'll come back to that, can drive senescence, as well as inflammation and damage-associated molecular pattern proteins and other molecules, for example, urate and so forth. There are at least 3 transcriptional pathways through which cellular senescence can develop. One of them involves P16 and retinoblastoma protein, the other P53 and P21, and there's a third sort of poorly characterized potential set of processes relating to inflammation. In culture, at least, it takes 2 to 6 weeks for a cell to become fully senescent. So senescence is a cell fate, like replication, differentiation, or apoptosis. A cell takes these multiple exogenous signals, integrates them, and makes a fate decision to become senescent, and then 2 to 6 weeks later, mature senescent cells can develop. And the rate at which they occur and develop depends on the starting cell type. Some, but not all, senescent cells can acquire a senescence-associated secretory phenotype that we heard about in the last talk. As we heard in the last talk, IL-6 is important in that set of processes. A couple of thousand genes have changes in their regulation as this develops. And there are changes in not only secreted proteins and peptides, but also there can be secreted nucleotides, including microRNAs, exosomes, and other and bioactive lipids and other molecules as part of the SASP. The SASP likely contributes to multiple aspects of tissue function, both locally and systemically. Now, senescent cells are resistant to apoptosis, and as we'll talk about later, this is important in targeting these cells. So we've reported at least 6 what we call SCAT pathways, senescent cell anti-apoptotic pathways, and 2 more are about to be published. As we heard in the last talk, senescent cells accumulate with aging in multiple tissues right through the vertebrates from fish up. And what's shown here is senescent cell accumulation in adipose tissue of healthy individuals who are donating a kidney. So this shows that senescent cell abundance is higher in older kidney donors than younger kidney donors. In most tissues, senescent cell burden remains relatively constant through the mid to late 60s, and then there's an exponential increase that occurs. This appears to be coincident with development of risk of frailty and other parameters, as I'll talk about later. So senescent cells can have a secretory phenotype. Here I'm showing the secretory phenotype that's characteristic of human preadipsites. The reason I'm talking about preadipsites will become evident later as we talk about development of senolytic drugs. Unlike some other cell types, senescent preadipsites express not only IL-6 and other interleukins, but also TNF-alpha. As components of the SASP, plus chemokines that attract various immune cells, extracellular matrix proteases, and other kinds of molecules like plasminogen activator inhibitor 1 that cause hemostasis. Very small numbers of senescent cells can cause problems. This shows what happens if senescent autologous ear fibroblasts, 200,000 of them, are injected around the knee joint of mice in the 2 lower figures, or if control non-senescent ear fibroblasts from the same mice are transplanted around their knees. And what we see occurring in a fairly short period of time, after a couple of months, is what looks almost like human age-related osteoarthritis, with thinning of the joint space, loss of cartilage, bony spicle formation, subchondral osteoporosis. So this shows that senescent cells can, if introduced into a young individual, cause dysfunction that resembles the kinds of disorders that are seen in older individuals. Indeed, very small numbers of senescent cells can do this. So here, what we're showing is that if 1 million senescent cells are transplanted into the abdomen of young mice, we see after a few weeks development of frailty with problems with gait speed, ability to hang from a wire, grip strength, and other parameters. Furthermore, lifespan is shortened in these young animals following senescent cell transplantation compared to transplanting non-senescent cells. In this case, the cells are made senescent either by radiation or by chemotherapeutic drug exposure. And the senescent cells that are transplanted are non-autologous preadipcytes because these cells don't induce as much immune response as other cell types, or autologous ear fibroblasts that are made senescent by radiation or not, or human preadipcytes injected into SCID mice. So in all of these models, we find that if only 1 in 10,000 cells in the recipient mouse is a senescent cell, this is sufficient to drive widespread dysfunction and early death. Furthermore, these small numbers of senescent cells accelerate onset of age-related diseases as a group. There's no one particular disease whose development is accelerated. And amongst the diseases that are accelerated in development by these small numbers of senescent cells are a variety of cancers. One of these senescent cells that were transplanted, some were tagged, so we could distinguish the recipient's own cells from the transplanted cells. And the transplanted cells remained within the peritoneum. Their numbers decreased over time, over a couple of months. But what we found was that the recipient's own cells became senescent, and this occurred everywhere in the recipient, even at a distance from the intraperitoneal area. For example, in the brain, in the limbs. So senescence can spread from cell to cell, and this can occur systemically. We set out to develop senolytic drugs back in 2005, and this was based on 3 important publications. One was by Eugenia Wang in 1995, showing that senescent cells, as I indicated earlier, are resistant to apoptosis. Indeed, you can grow senescent cells under conditions that would kill normal cells in a few hours for up to years. In 2004, Ned Sharpless, who's now director of the National Cancer Institute, came out with an important paper in Journal of Clinical Investigation that showed senescent cells accumulate, but the rate of accumulation of senescent cells can be decreased by interventions that enhance health span. For example, caloric restriction, or in Ames dwarf mice that have deficient growth hormone production and extended lifespan. And then also Judy Campisi published in 2008 the senescence secretary phenotype, but she mentioned this in meetings well before that. So this led us to begin efforts to try to develop senolytic drugs in 2005. This was a slow process. We tried using fusion proteins that had a killing construct and something that would home the cells to senescent cells. Didn't work. High throughput screening, et cetera, et cetera. And then finally, we decided on using a hypothesis-driven approach to try to develop drugs that would selectively induce death of senescent cells, what we call senolytics. We asked why senescent cells are resistant to apoptosis, as Eugenia Wang had found, despite the fact that they are causing apoptosis in surrounding and distant non-senescent cells. So this led us to ask whether there are pro-survival pathways that defend senescent cells against their own pro-apoptotic SASP. We also reasoned that senescent cells are in many respects, metabolically and otherwise, similar in some ways to stage 0 cancer cells. We use those 2 hypotheses to develop the drugs or potential drugs in silico. And it actually, once we settled on these hypotheses, it only took a few days in May 2013 to come up with the drugs. So we discovered that there are senescent cell anti-apoptotic pathways, as I mentioned before. These pathways are more networks than pathways, and they interact. Different senescent cells types depend on different pro-survival pathways to defend themselves against their SASP. And these differences could be related to differences in the SASP across different kinds of senescent cells. So, for example, endothelial cells, human endothelial cells, depend largely on BCL2 family members to protect them from their own SASP, whereas, for example, fat cell progenitors or preadipcytes depend on several SCAPs that are redundant. If you target just one of them, you will not kill the cells. So we verified the importance of these pathways using RNA interference approaches, siRNA screening, and we found the key nodes on these SCAT pathways, and then we used various programs, including CMAP, to try to discover drugs and natural products that are known to target the key nodes on the pathways, and we combine some of these or use drugs that hit multiple targets in order to develop senolytic strategies. So the first two drugs that we discovered are senolytic are dasatinib and quercetin, and they're mainly effective in combination. Quercetin on its own will not kill a lot of senescent cell types, and there are some senescent cell types that dasatinib on its own will not kill, but when they're combined, they do. Dasatinib is a tyrosine kinase inhibitor. It's used for treating certain kinds of tumors. It's not a chemotherapy drug in the usual sense. Related pan-tyrosine kinase inhibitors do not work, like imatinib. So there are small changes you can make in senolytic molecules that have a big effect on their senolytic activity, which has led us and others to do a lot of medicinal chemical optimization of these compounds. We find that different senolytics, because they target different SCAT pathways, will target different kinds of senescent cells. We have not found or seen any published senolytic so far that targets absolutely every kind of senescent cell, including the FOXO4 related peptide. So we can find examples of senescent cells that each drug will not target, and this arguably is because of the different SCAPs that different kinds of senescent cells employ to defend themselves. So far there are about a dozen drugs that have been discovered. They all operate on this on these SCAPs. That's how they operate so far. There are additional drugs that are being developed that target where surface receptors are targeted by vaccines or toxin delivery approaches, and there are immunomodulatory approaches which are being taken to try to enhance immune system clearance of senescent cells. Senescent cells in vivo are killed by immune cells. They are resistant to other forms of cell death. So in order to test these kinds of drugs, we transplanted luciferase-expressing cells into the peritoneum of mice and then treated with senolytics or control agents. And the senolytics specifically targeted the transplanted senescent cells, but not transplanted non-senescent cells. Most recently, we've, in the first human study of senolytics, that was reported in July, we took adipose tissue right out of the operating room from obese diabetic individuals who'd given informed consent. In obesity and diabetes, there's a large accumulation of senescent cells within fat tissue. So these explants were immediately placed into a solution of either senolytics or vehicle, and within a few hours, the senescent cells were dying, but non-senescent cells did not. So this shows telomere-associated foci, which is one of the better markers of senescence, but we used many other markers, including P16, to show that senescent cells selectively killed senolytics selectively killed human senescent cells in these explants. And they did so through apoptosis. This shows that cleaved caspase 3 is increased in these adipose tissue samples containing senescent cells treated with the combination of dasatinib and quercetin compared to vehicle. In the medium from these cells, we found decreased production of SASP factors and increased production of beneficial adipokines such as adiponectin. And we also found evidence that adipose tissue stem cell function was restored and insulin responsiveness was restored. So in vivo, this shows some of the first studies we did with the original senolytics, dasatinib plus quercetin, given once monthly or every 2 weeks in this case. This shows what that there's improvement in frailty in prodroid mice. These are the ERCC1 minus delta mice that Laura Niedernhofer had developed and collaborators. We found that there's decreased sarcopenia and neurologic dysfunction, so there's less splaying of the legs in these animals than in vehicle-treated animals. And there were many other phenotypes that were alleviated, including in chronologically aged animals. We found an increase in cardiac ejection fraction and other parameters. Very recently, a paper came out from colleagues of ours in San Antonio in Aging Cells showing that Senolytics alleviate Alzheimer's-like changes that occur in 4 different types of tau-positive mice. So in this case, the combination of dasatinib and quercetin was given every 2 weeks for 3 months. Incidentally, the elimination half-life of these drugs is about 12 hours. Because senescent cells take 2 to 6 weeks to form, these drugs can be given intermittently in a hit-and-run approach. We're not trying to continuously occupy a receptor or affect an enzyme. So this treatment reduced neurofibrillary tangles, a decreased ventricular enlargement, actually partially reversed it, improved or reduced neuro degeneration and neuroinflammation in 23-month-old tau-positive mice. Targeting senescent cells in bleomycin-induced pulmonary fibrosis, either in intact mice using AP20187 or in wild-type mice using dasatinib and quercetin given intermittently, reduced the problems with lung compliance that these animals have, attenuated the degree of loss of body weight, and improved treadmill endurance. This has led to clinical trials that are now underway in idiopathic pulmonary fibrosis in humans. This is a senescence-driven disease. There are few treatments that have any kind of effect at all, and really the only disease-modifying treatment is lung transplantation, which of course you can't do in elderly people. So this could be an early indication for these agents, depending on how the clinical trials go. I mentioned before that intermittent treatment appears to work with Senolytics. As you'd expect, they take time to appear. If you give a single stimulus to induce senescent cell formation in young mice, for example, a blast of radiation to one leg, such that the hair turns gray and the animals have trouble walking, a single dose of Senolytics, or a single treatment course for a couple of days of Senolytics, is sufficient to restore these animals' ability to walk, and this persists for months. And indeed, we found that the single-leg radiated animals had a shortened survival, and that was in unpublished work, and that was corrected by a single dose of Senolytics 3 months after their radiation exposure. I'll skip past to the next slide. We heard a discussion about bone before. Amongst other cell types that become senescent-like in old animals in their bone are in fact osteocytes. So it's turning out that a number of non-dividing cell types can acquire senescent-like phenotypes. In addition, there are senescent cells that form in the bone marrow. If we use dasatinib and quercetin once a month for 4 months in old animals, we find that we're able to increase cortical bone thickness and trabecular bone in the femur, trabecular bone in the spine. And the mechanism is by decreasing function of osteoclast progenitors and increasing function of osteoblast progenitors, so that we get a decrease in bone resorption, which is the main effect of senolytic drugs, resulting in bone reformation. But there is also an increase in new bone formation as well. So unlike the currently available treatments that decrease bone resorption, there is no uncoupling that occurs. There is no coupling that occurs between decreased resorption and formation, so that we're able to decrease both resorption and increase formation with these agents, leading to, again, clinical trials. Senolytics reduce endomal calcification in ApoE knockout mice. One of the common things that we see in older people is arteriosclerosis. This is where you see calcium deposited in the large blood vessels associated with decreased vascular distensibility and systolic hypertension, a major cause of strokes. There's no really good treatment for this loss of distensibility of the aorta and calcification of the aorta. unlike atherosclerosis, for which there are multiple treatments. In mouse models of calcific sclerosis of the media of blood vessels, we find that senolytics reduce bone formation, reduce calcification. So one of the things that senolytics seem to do across different tissues is they result in less of a tissue where it shouldn't be and more of a tissue where it shouldn't be. In other words, a reversion to a sort of youthful pattern. So we've decreased effectively calcification and, in a way, bone formation in the aorta and increased bone formation in bone. So I mentioned before about the transplanted animals. If we treat the young animals that are transplanted with senescent cells with senolytics, either at the time of transplantation or a few weeks later, we prevent the frailty that occurs. We also found that in very old mice, if we treat with senolytics, we reduce frailty. So here I'm showing what happens in 20-month-old mice treated once per month. They have improvements in walking speed, grip strength, wire hanging, and so forth. These animals also have an increase in remaining lifespan of 36%. So if you treat very old mice, 24 to 27-month-old mice, with senolytics once a month, this results in an increase in remaining lifespan. And another senolytic that we're about to publish with collaborators at University of Minnesota does the same. And again, as occurred after the transplantation, we found that all age-related diseases are delayed as a group in these old animals that are treated with senolytics, including cancers. So there's an increase in remaining lifespan, but it's not at the expense of living in a frail condition. There are many other conditions that it looks like senolytics may have a role in, or senescent cell clearance. So there are studies going on giving some, as you'd expect if you're targeting a fundamental aging process, there should be multiple downstream effects. There are a number of ways that these drugs are being taken through to the clinic now. I mentioned that there are a lot of different senolytic drugs. They seem to work in different conditions to a different extent. Here we've got 6 clinical trials underway for various conditions, and various other groups have clinical trials underway. So the initial clinical trials are for individuals with very serious conditions where the risk-benefit ratio justifies proceeding with the trial, and also we have to, we feel ethically bound to demonstrate that there are senescent cells in the subjects with the particular conditions before we enroll them in the trials. So these are early proof of concept kinds of trials that are underway, and really when you get to people, it's really difficult to predict what will happen. We don't know what all the side effects of these of senolytics might be. Different individual senolytics can have drug-specific side effects, but we don't know much about class-specific side effects yet, and we need to figure that out. So in conclusion, it appears that, and I didn't have time to go into it, but across different organs, including the brain, including fat tissue, including bone, one of the ways that senolytics appear to work in clearing senescent cells is to alleviate stem and progenitor cell dysfunction. And this is usually related to where we've done co-culture studies and looked at knocking out particular SASP elements based on that. Typically, 3 to 5 SASP factors explain around 50% of the effects on different kinds of stem cells, depending on the tissue. We find that senolytics delay or alleviate chronic diseases and frailty and enhance health span and remaining lifespan in mice. It looks like intermittent treatment is as effective, if not more effective, than continuous treatment because we're giving these drugs in a hit-and-run approach. And it looks like these agents might lead to interventions in humans that could delay, prevent, or alleviate senescence and age-related conditions as a group. But we have a lot of work to do on the translational side to determine if these drugs are effective and safe. And nobody should be taking these drugs until those kinds of trials are done. And I'll just conclude by thanking all the people who've done this work and the various funding groups who've helped us out. Thank you very much.
Thank you so much, Dr. Kirkland, and actually thank you to both of our speakers for the fantastic presentations. We are running a little bit over time, but hopefully our audience can stay with us, and the speakers can as well, just to answer a few questions that we've received. If you do have any further questions, please feel free to submit them. I can't promise we'll get to them, but I'll do my best. The first question actually came in a number of different forms, and I'm going to put this to both of you. I'll start with you, Dr. Kirkland. Given the tumor-promoting effects of senescent cell accumulation and some of the other effects that we see, why is apoptosis not favored by selection as a more favorable form of cell termination? And are there any advantages that you can hypothesize to having senescent cells around that outweigh the increased risk of cancer and other late-stage health risks?
Well, in younger individuals, and in it appears that senescent cells are a cancer protective kind of mechanism. So, if they're potentially oncogenic insults, first of all, if cells enter a senescent cell, they stop dividing, and then they kill the cells around them. So if there's a developing clone of cancer cells, these cells will kill the cells around them, and then they're eradicated by the immune system. So they attract, activate, and anchor immune cells. So that could be the beneficial side. The view is if they achieve a certain threshold, they may promote cancer development for the reasons that Sheila mentioned, and also they do things like, in addition to producing IL-6 and other things that could promote tumor development, they produce proteases that can break down the extracellular matrix and allow tumors to metastasize more readily, and they also produce factors that impair immune function. So IL-6 actually, if it's high enough, will prevent macrophages from migrating, and then some of the proteases these cells produce will cleave off fast ligand and other kinds of things that immune cells depend on. Furthermore, what happens when you get to a certain point with respect to senescent cell abundance, senescent cells induce senescence in other cells. So we find that in young animals, there's a threshold of 1 in 10,000 cells that has, if we inject 1 million cells into a young mouse, will cause a downward spiral. In an older mouse, we only have to inject 500,000 cells. So there appears to be a point where the formation of new senescent cells caused by existing senescent cells outstrips the ability of the immune system to clear senescent cells. And furthermore, once there are enough senescent cells around, they begin to poison the immune system, and the immune system may undergo other kinds of aging changes itself. So there's a counterbalance between spread of senescence and ability of the immune system to clear senescent cells, potentially explaining this exponential rise you get later in life.
Thank you. Dr. Stewart, did you have anything you'd like to add?
I think that was a great explanation, but I think one thing that is always confusing for people and worth pointing out is something James mentioned, which is, my 25 minutes talking about the tumor-promoting events of senescence, but, it was originally described as a tumor suppressive mechanism, and the answer is, can you have both? And the answer is yes, right? So, from an evolutionary standpoint, if you can keep your organism cancer-free until its babies are self-sufficient, which is where we don't see cancer, and it comes at a cost later, this is something Dr. to discuss as antagonistic pleiotropy, it's very, it's blind to it, right? So it does play a potent tumor suppressive effect when it is in a cell autonomous setting. And one other thing I'd just like to point out is one of the things that we see, and this is really interesting to look at, is a senescent cell is not a senescent cell. Again, James brought this up. What different types of senescent cells express seems to be quite unique to the tissue from which they arise. And the immune system certainly can clear these cells, but often when it's fibroblasts, they don't. So I think there's a lot we still don't understand and quite a bit of room for investigation.
Great. Thank you so much, Dr. Stewart. Another quick question for you. Would you expect a response in humans to p38 inhibitors to be broad, or are there markers that you might look for that would predict an individual response?
At this point, looking at the models, we seem to affect both activated stromal cells as well as what we see with senescence. We often like to think of senescence as another way to get the stromal phenotype. So that I don't know the answer to that question yet. We're certainly looking at it, but I do expect it to be broader than senescent cells for sure.
Great. And Dr. Kirkland, just a final question for you. You mentioned, I think you mentioned this briefly in one of the slides of your talk, but this viewer asks if there's any senolytic compounds that target unique characteristics of senescent cells apart from apoptosis resistance.
Yes, there is work going on, most of it unpublished, using immunomodulators to try to enhance immune cell clearance of various kinds of senescent cells. There are groups and companies who are developing fusion proteins and proteins that are attached to nanoparticles that carry cargoes that kill cells that they encounter, and with the other end of the fusion protein carrying a domain that recognizes senescent cells. There are vaccination approaches that are being developed, and then there are drugs that are being developed that target the metabolic phenotype of senescent cells. So I mentioned before that senescent cells really are like cancer cells. It can't divide in many ways, so especially from a metabolic perspective. So they have a partial Warburg shift, much like cancer cells. And it appears that certain there may be ways to target senescent cells because of their dependence on glucose metabolism as opposed to fatty acid metabolism. So there are a lot of ways that are being developed to try to target senescent cells. In addition, drugs that target the secretory phenotype of senescent cells, and those include the P38 inhibitors that we heard about before, but also JAK1/2 inhibitors, drugs like ruxolitinib, drugs that interfere with protein production like rapamycin, and drugs that interfere that target mitochondrial complex 1 like metformin, and some new drugs that are much more effective, thereby increasing AMP kinase and inhibiting mTOR. Those drugs actually, it looks like, prevent spread of senescence. So although those drugs have to be given more continuously than senolytics, they can actually result in a decrease in senescent cell burden because there's this tension, as I mentioned before, between new senescent cell generation because of existing senescent cells spreading senescence on the one hand, and the immune system clearing senescent cells on the other.
Fantastic. Well, unfortunately, we're going to have to end things right there. It just remains for me to thank our speakers, Dr. Sheila Stewart from Washington University in St. Louis, and Dr. James Kirkland from the Mayo Clinic. Please go to the URL now at the bottom of your slide viewer to learn more about resources related to today's discussion, and look out for more webinars from Science, available at webinar.sciencemag.org. And be sure not to miss our final installment in this series to be broadcast on November 14th. This webinar will be made available to view again as an on-demand presentation within about 48 hours from now. And if you'd like to receive alerts about future webinars, please go to the sign-up link in the Resources tab just below the slide window. We are interested to know what you thought of today's webinar. Please feel free to send us an email at the address now up in your slide viewer, webinar@AAAS.org. Again, thank you so much to our panel and to Cell Signaling Technology for their kind sponsorship of today's educational seminar. Goodbye.